tiene las fuentes, los tipos de letras que no hay en la literatura, la computadora acá, entonces se cambia un poco. No,
Hola. Hola, buen día a todos. Bienvenidos. Este es nuestro primer seminario de este ciclo, de este semestre. Entonces les damos la bienvenida a los que están de forma presencial, pero también a todos los que nos siguen a través del canal de YouTube del Instituto de Biología. En esta ocasión tenemos el placer de contar con la presencia del doctor Bernardo Santos, del cual voy a hacer una breve descripción ahora. Él es investigador del Center for Integrative Biodiversity Discovery del Museo de Historia Natural de Berlín. Es originario de Brasil y cursó sus estudios de licenciatura y maestría en la Universidad Federal de Espíritu Santo. Hizo su doctorado en el Museo de Historia Natural de Estados Unidos y cursó 4 años como investigador postdoctoral en la Smithsonian Institution. Fue curador de himenópteros en el Museo de Historia Natural de París de 2020 a 2022 y su investigación se centra principalmente en la comprensión de la sistemática y la evolución de avispas parásitas, utilizando un enfoque en el que integra aproximaciones taxonómicas, genómica, análisis cuantitativos del fenotipo, así como estudios faunísticos y de historia natural. Bienvenido, doctor Bernardo. Entonces, le cedemos la palabra.
gracias por la introducción, gracias por todos de estar en acá. Mi español no es muy bueno, entonces No tengo practicado mucho, pero voy entonces hacemos así, voy a presentar en inglés, pero si tienen preguntas y quieren hacerlas en español, yo hago mi mejor para responderlas bien así. Okay, yes? All right. So yeah, thanks again, everybody, for being here. Can you hear me all right? Yes? Okay. So yeah, I'd like to talk to you a little bit today about some things in my trajectory and my research that I think are interesting for other people who also, like me, have a background on systematics and taxonomy, but face the situation of having that kind of research not always extremely valued in science, and having to look to other kinds of projects as well to try to valorize or to put more value into their CVs and things like that. And at the same time, how these things can really connect to our goals and our interests in systematics and taxonomy. So as mentioned in this very nice introduction, I'm originally from Brazil, from Vitoria. I did my undergrad and master's there, but then I went to the US to do my PhD and postdoc, always in natural history museums. Then I became the curator for Hymenoptera in the Paris Museum, and now I have just joined the Museum für Naturkunde in Berlin. So I've traveled quite a bit, but always working in natural history museums and collections work, and always working with parasitoid wasps. So if any of you haven't heard Alejandro talk about it enough times, parasitoid wasps are insects that are free living as adults, but they lay their eggs in the bodies of other insects, their hosts, and their larva then develops by slowly consuming the hosts, eventually causing its death. So parasitoid wasps are extremely diverse, and specifically I work on the family Ichneumonidae, which is the largest family of parasitic wasps, with over 24,000 described species, but we estimate that there's over 100,000 still waiting to be described. And the Ichneumonids are very cool wasps. They can be very colorful, big or small. They're found in all ecosystems all over the world. They're super abundant and do lots of interesting things, as we will see. And normally, as I said, I started my career, my trajectory as a taxonomist, essentially, someone who was interested in going to the field and collecting specimens, looking at collections of pin specimens, of specimens in alcohol, sorting specimens, preparing material, and then making this into revisionary taxonomic works, or also in phylogenetics, in which I was interested for like classification's sake. I was interested in phylogenetics because I wanted to know how these species related to each other and how that could inform classification. So just standard systematics stuff. And I did that for several years in Brazil. And then interestingly, when I went to do my interview for the PhD program in New York City, I was talking to some of the other students in the program, and they were asking me what I was interested in doing. And I said, well, I'm interested in doing a phylogeny of the subfamily of Ichneumonids. It's a really big group. I think it would be very important to do a phylogeny to understand their classification. And what several different people told me there was, well, that's not enough. You can't just do a phylogeny just because you're interested in the classification. That's not going to get you anywhere in your career. You need to have a sort of like evolutionary question that you're interested in answering with your phylogeny. If you're just doing systematics, it's going to be really hard for you to find, a fellowship, for you to find a postdoc, for you to find a job. So you need to frame your work in sort of such that you're answering an evolutionary question with your phylogenies. And for taxonomy and species descriptions, just forget about it. Do it in your spare time if you can, but don't expect that to bring you any kind of advance in your career. So I was pretty bummed out with that, because I was pretty sad, because Ichneumonids are, as I said, super diverse, and there's so many species to be described, right? We have been quite bad about it in the past few decades. Here's the number of species of Ichneumonids described per year across the past century or so. And you see that we're not improving, we're not getting better at this. We keep more or less at the same average of 212 species per year. And in that pace, if we estimate that there's about 100,000 species to be described, it's going to take us 450 years to finish. So that's not great. And on top of that, right, the resources for taxonomy are dwindling. As I was saying, I was getting advice from many people saying, there's no point if you just do taxonomy, you'll never get a job, you'll never do, you never be able to do a career by just doing taxonomy. And as we know, nowadays there's many people who say, taxonomy is too slow. The only way to advance in our understanding of biodiversity is that we should do these minimalistic approaches to taxonomy, where we just sequence one gene CO1 and do DNA barcoding, and then one DNA barcoding cluster is going to be equal to one species, and that's it, so that we don't bother, taking all that time to describe species. So it's a difficult scenario, right? But I knew about that, but I also wanted to do a PhD, and I wanted to get a job. So I actually heard the advice that people told me, and I did as they told me. I started to try to diversify my research program so that, I was working not only on wasps and new species and revisionary taxonomy, but also trying to understand some aspects of evolutionary biology, of ecology, and this kind of like big questions. So what I want to talk to you about today is just a little bit of this work that I've done, how I went out of taxonomy, so to speak, for a while to do other things, and how in the process I realized that a lot of these other things actually have a lot to do with the kinds of data that we generate in systematics and the kind of questions that we're interested in, and can kind of like fit in a research program that incorporates biodiversity discovery and taxonomy, which in the end I believe is a unifying principle of biodiversity research. And we can strive to incorporate these elements into our systematics work, I hope. And ichneumonids are actually a really good system for investigating evolutionary questions, because they do a lot of interesting things. They parasitize a very big diversity of hosts. They're all parasitoids, but they attack, some of them will attack Lepidoptera, some will attack beetles, some will attack other wasps. They have a diversity of hosts and lots of interesting behaviors. So for example, some species, they will attack hosts that are hidden inside of a substrate, inside of wood, or inside of soil or something like that. So they developed forms of echolocation in which they tap the surface of the substrate with their antenna and use the echo from that the sound clues that they get from that with specialized hearing organs to find out where the host is, and then they insert the ovipositor into the wood and sting the host and lay the eggs there. Some other species of ichneumonids, they've developed a symbiotic relationship with viruses. So they inject viruses along with the eggs on their hosts, and these viruses will help to destroy the immune system of the host. Other ichneumonids, they can change the behavior of the host. So here is a ichneummonid larva attached to a spider. And as the ichneummonid slowly eats the spider alive, it will change its behavior. So the poor spider will go from building a web, a normal web that is designed to catch prey, to a web that is actually designed to protect the cocoon of the ichneummonid, of the wasp, after it kills the spider and starts its metamorphosis. So it's going to entirely change its web, so to build a protective encasement for the wasp. So all of these interesting things that these wasps do, and that make for a great subject for investigating their evolutionary origin, how many times these behaviors evolved, and so on. So essentially, this element of my research program is what I call the effort to link form and function. We most of the time we have these we might have these behaviors, but we don't observe them in nature, right? Most of us, if we're systematists, we collect these insects in traps and we never see them doing anything. We just see them dead in a drawer or in a box. So what we see the most is some morphological characters or phenotypic characters, right? So what we want to understand is the link between these cool behaviors, between the ecological traits and the phenotype. We know, for example, that obviously what the species do can influence their phenotype, right? Like their behavior, their ecological traits will shape the morphological traits that we see. But it also goes the other way around, right? Because certain phenotypic traits, they can constrain what kind of ecologies these organisms what kind of ecological roles they can play. And in the middle, between these two things, we have phylogeny, which works as sort of a mediating effort, element between ecology and the phenotype. So when we start to look at all these three things combined, there's all sorts of interesting questions that we can ask from our data. So for example, do ecological shifts show phenotypic signatures? Do we see changes in the phenotype when we see changes in the ecology? Can we predict ecological traits from the phenotype? Can certain traits accelerate diversification, et cetera, et cetera? So there's all these interesting things. And what is cool about it is that to investigate these questions, there's two main things that you need. You need one, phylogenetic trees, which is something I already do for my normal work as a systematist. Look at that. And we need phenotypic data. You know, we need measurements or morphological characters coded as zeros and ones, or photos, or any kinds of things that we can quantify the phenotype. And that's also something that we do all the time as systematists, right? We're interested in looking at characters, collecting information about specimens. So in the end, what we need are two things that as systematists, we do all the time anyways. So for example, one quick example of something I was interested in was convergent evolution. So in ichneumonids and the specific subfamily called Cryptini, as I said, there's many species that attack hosts that are hidden, concealed in substrates. So here's a wasp attacking a beetle larva that lives in wood, and here's a wasp, an ichneumoniid that is perforating a nest from a predatory wasp, a nest made of clay. So two examples of wasps that they're attacking hosts hidden in deep substrates. And what I saw is that they develop similar suites of traits, of morphological traits to do that. Even if they're attacking different hosts, the fact that they're all hidden and concealed, they develop these hammer-like antennal tips to tap the surface to produce pulses of sound. The swollen foretibia because it houses the specialized hearing organs. We have stronger oviposition muscles. We have strong mandibular muscles to emerge from the substrate. So I started to look at all these characters. And I saw that they, you know, when you have the phylogenetic tree for the whole subfamily, they evolve several times. I'm not sure if you can see here the little pink branches in the tree, but you see that these traits, they evolve several times. So independent of the lineage, you always see these same traits evolving when the wasps are attacking deeply concealed hosts. And they're also correlated to each other and correlated to the use of deeply concealed hosts. And so this was an interesting example of how ecological traits can be associated with specific phenotypic traits. But sometimes there's something about the specimen that we look and we cannot quite translate into like a 0 and 1 discrete trait, right? So we're interested also in looking at phenotypic traits. They're not discrete, they're not zeros and ones. And one of the tools I found that really helps for that is geometric morphometrics, which is the statistical study of shapes. So in this case here, for example, we have a species, and I've defined 6 specific locations in the mesosoma, in the thorax of this specimen, that I think are kind of indicative of the overall shape of the body. So we can take a standardized photo, always in the same position. We place these landmarks in specific areas of the body. We then discard the image information and we keep only these landmarks. Then we go to another species and we do the same thing. We take the photo, place the landmarks, get rid of the image and keep that. But of course, we need to standardize these landmarks because they are often the wasps are often in different positions or alignments. They're different sizes and so on. So we use a procedure called a generalized procrustes analysis that will take these two things, scale them by size so that they're exactly in the same size. Then we will establish a centroid that is the middle point between all of the landmarks, align those, and then rotate them until we find the place where there's the least distance between them. And so with that, the landmarks are considered aligned and we can actually compare the shapes of two specimens, which is what we want. You do that for hundreds and hundreds of species. You align those, and then what you have is actually X and Y coordinates for each of the landmarks that can be treated as any kind of multivariate data. So you can do things, for example, as a principal component analysis. So here, each dot here is a species, and the closest the dots are to each other, the most similar the species are to each other. And so here, for example, in the first axis of this PCA, The first axis of variation here is describing an axis that goes here in red from very elongated species, so kind of flattened body, to very bulky, chunky species, robust species. And so you have different aspects of the variation that we can quantify and see in these graphs. And so we can start playing with things and saying, okay, where do we find, we have species that do this. Where do we find them in the graph? Are they all over the place, or are they concentrated in a specific region, right? This statistical rendering of the variation is what we call a morphospace. So you can see you can start asking things like, okay, how much of the morphospace is occupied by species that live in caves? How much of the morphospace is occupied by species that attack this kind of host, and so on. And then we can also link that with phylogeny to try to understand some aspects of diversification. So, for example, I was interested at the time at this one subfamily, this tribe called Cryptinae, and one element of my PhD was doing a phylogeny for this group. And what I found was that there were two major clades in the group. One of them was called what I call the Gabunia group, after the genus Gabunia. And all of the species in this clade here in green, they have this particular ecology in which they attack deeply concealed hosts. They go after hosts that are in wood or in nests and so on, and attack them, perforating the substrate with the ovipositor. So the kind of ecology that I just discussed previously. And the other big clade, what I call the Cryptus group, is a group that is much more diversified in terms of morphology and also in terms of biology. They attack many different kinds of hosts. So what I was interested in in looking was, well, the obvious hypothesis is that because this group has a conserved biology, they all attack the same kinds of hosts, we probably can imagine that they have lower rates of morphological evolution, right? They don't change a lot because they always attack in the same host, while this group will have lots of change. And so what we can do is that we can plot the phylogeny in our morphospace, creating what we call a phylomorphospace. So here the lines here between the dots here again are just our morphospace, right? The species and how different they are from each other. And the lines here represent the phylogenetic relationships between groups. So in red here, we had that, what I call the Gabunia group, just the group that has all of them have the same biology. And we do see that they are constrained to a specific area of the morphospace, right? They're just in one area of the morphospace. But when we measure the morphological branch lengths that separate each species, they are not shorter. So what we saw was that these species are actually changing quite a bit, but they're changing inside of a specific area of the morphospace. So the difference here, the difference in morphological diversity that we're seeing here is not because one of the group is not one of the groups has a lower rate of evolution. It's just because it evolves a lot, but only within a specific area of the morphospace. It's what we call running in circles in the morphospace. So it really shows how there's two different mechanisms for producing low morphological diversity, and also how there's not low morphological diversity does not equal stasis or lack of change. So I thought that was interesting and shows how we can use these methods to really try to understand what's going on in diversification in hyperdiverse groups for which we don't necessarily know a whole lot in terms of their biology. We can use geometric morphometrics for other things as well. So one of the studies that I'm just finishing now with some collaborators and two of my students is working on sexual dimorphism of in the subfamily Ichneumonidae, which is a big subfamily in ichneumonids that often has a lot of sexual dimorphism. So here's the female, here's the male. You can see that they're quite different from each other. For taxonomists, this is usually a nightmare because often you cannot easily associate the males and females of the same species. So one thing that we were interested in asking, besides, we wanted to quantify sexual dimorphism, seeing which species we have more or less dimorphism. But also one interesting thing is, how is dimorphism structured? So is it structured in a predictable way? For example, here in this graph, let's say that here we have males and females, and always, the females are located to the left of the respective males, right? That is to say that the males are always a modified version of the females, that they're always, different from the sexes are different, but always in the same way. And another possibility, for example, is that actually we have the females scattered all over the morphospace, reflecting, for example, their need to attack different hosts, and the males actually show a generalized shape, and they're located in a specific area of the morphospace because the males in parasitic wasps don't really need to do much. They only need to go after the females, and it's the females who are doing all the specialized ecological stuff because they're going after the hosts. So I wanted to look at these questions, and also maybe instead of either of these patterns, what we see is we see more dimorphism in species that have a particular ecological trait. Species that do something specific have more or less dimorphism. So I recruited some students back in Paris to work on this, and what we did also some colleagues at the Utah State University who took a bunch of photos. So they took, again, photos are something that we use all the time for systematics work, right, for taxonomy. So it's kind of our normal work life. So what we did was that we selected 24 species in Ichneumonidae, and we took photos of 10 males and 10 females of each species, both of the head and the mesosoma, and we put landmarks on them. And then with that, we could quantify the sort of degree of sexual dimorphism, how different males and females are across each of these 24 species. And with that, we looked at a bunch of different things, but what I'm interested in talking about here is that we saw, for example, that in terms of the hypothesis I was talking about, it seems that was true. They see when we compare the males and the females of each species in the morphospace, the difference seems to be structured in sort of like a predictable way. So if you look at the lines separating the males and females of the same species, in most of the cases, they're going in the same direction, right? So we did statistical testing of this. And in 78% of the cases for the head and 100% of the cases for the mesosoma, the change in morphology that we observe between males and females is consistent. It's predictable in some ways. So that was interesting. And at the same time, we saw as well that species that attack pupae, they tend to have more dimorphism, especially for the head. So we got species that are some species of ichneumonids, they oviposit in the larva, and others will oviposit in the pupa of the host. And especially for the head, here on the left, we have the head, and here on the right, we have the mesosoma. But especially for the head, I think you can see clearly that the species in red here that parasitize pupa, they tend to have more dimorphism than the species in blue that parasitize larva. We do, for example, have three examples of species that parasitize larvae and have high dimorphism. But in all of these cases, what's interesting, well, let me go back a second. What is interesting is that species that parasitize pupa, what we hypothesized is that they have higher dimorphism because the pupa are often hidden. They're often buried inside of the soil, especially Lepidoptera and pupa. They're often buried inside of the soil in pupation's chamber. So what we thought was, well, then the female needs to dig to look for the pupa, and the male doesn't need to do that. So the species that attack pupa, in those species, the female, we have special adaptations in the head, and that's what makes it so different from the males. And we do have 3 species that attack larvae that have high dimorphism. But what's interesting is that in all of those cases, they attack larvae that are also hidden or buried or inside of stems and that kind of thing. So it kind of, even if it's contrary to the larva pupa distinction, it also goes in favor of our hypothesis that species in which the female needs to dig or find the host in some different way will have more dimorphism than the species that in which the host, that attack caterpillars, they're just out in the vegetation and it's exposed. So these are just, some examples of how we can use geometric morphometrics to look at interesting traits for ecological traits, and they're linked to some interesting evolutionary attributes. I'm also interested in phylogenomics, of course. As I said, I never cease to be a systematist. So I'm doing work with ultra-conserved elements, which are parts of the genome that are conserved across a broad spectrum of taxa. So you probably heard lots about UCEs already. I know that there's lots of people here at UNAM who work with these methods, but I've been using them a lot for generating phylogenies of ichneumonids. So, for example, here's at this point, we're at the point where we have 800 species of ichneumonids in our tree, with big data sets with over 1,000 loci per sample. What's interesting about UCs too is that you can generate data for really old specimens. So you're not really constrained to what you have just collected, or there's really fresh material in freezers, etc. You can go to the museum and sample a specimen that's 70 years old and still get data. And then, it's cool because now we have this big tree again, once you have this, besides all of the implications for taxonomy and systematics, you can start thinking, well, what we can do with it, what kind of questions we can ask that can be answered with these big comprehensive phylogenies. So I started to think about whether we can extract some information about ichneumoniid diversification and how it links to some interesting traits. This is challenging, of course, because it relies on a bunch of statistical modeling that is often difficult to validate, right? So we don't know for sure that we can produce reliable time trees. Diversification dating is a work in progress, and it's something that where methods are still being actively developed, and there's a lot of debate on how reliable they are. Can we infer complex evolutionary events from phylogenies? How much we can really tell just from these branching trees what has happened in the past, right? But I was interested in doing some exploratory work and seeing what we could find. So one thing I decided to test was this very interesting divide in ichneumonids that we see in ichneumonids between what we call idiobionts and koinobionts. The name is complicated, but it's actually quite simple. Idiobionts are parasitoids that they will sting the host and paralyze it and stop their development. So the host will be just paralyzed for the whole time, and will be just a bag of meat that the host will eat slowly. And koinobionts, on the other hand, are the more kind of stereotypical parasitoids in which they lay the egg, but they don't permanently paralyze the host. So the caterpillar or whatever the host is keeps eating and developing while it's being parasitized. This seems like a trivial distinction, but it actually is correlated and linked to a bunch of other ecological traits. So idiobionts are, for example, usually ectoparasitoids. They're usually more, that is, they attach to the outside of the host. They're usually generalists, in that they attack lots of different species of hosts. They usually attack pupae, while koinobionts are usually endoparasitoids. They're much more host specific because they need to deal with the physiology of the host more closely because they're inside their body. They often can attack earlier developmental stages, so they often attack larvae. And so there's this hypothesis in the literature about ichneumoniid diversification that says, well, in idiobionts, because in idiobionts, the differences between species are usually morphological because they reflect how they need to go to look for hosts in different environments. Whereas in koinobionts, the differences between species are usually physiological because they reflect the need to deal with the immune system of the host and with the specific physiology of each host. So I thought, well, that's interesting. It kind of intuitively makes sense, but nobody ever really tested that, right? So I thought, well, what we would see, I imagine, is that now we have this big phylogeny. We can start to test it. We have idiobiont species. We have koinobiont species. What we would see is that if in idiobionts the differences between species are more morphological, we would see higher rates of morphological evolution in idiobionts, right? We would see that when we plot them into the morphospace, they change more than the koinobionts. So I recruited Jean Colaza, who was a master's student back in Paris, to work on this issue. He was a very good student, very smart with R and statistics. So I said, let's tackle this project together. Luckily, when I was doing this big phylogeny of ichneumonids, I saved all of my voucher specimens, made a really nice voucher collection. And what we did was that we photographed every single specimen. This also helped us to check identifications, where whenever I was not sure about the idea of a specimen, I could send the photos to colleagues and ask what they thought. But of course, this is also material for geometric morphometrics. So again, we use those photos to put landmarks and generate geometric morphometrics data, which Joan analyzed. So the same thing that you already saw, we used this as multivariate data and combine it with the phylogeny. And what we saw in the end was quite different than what I expected. The results were reversed. The diversification rates were much higher in koinobionts than in idiobionts, both for the head and for the mesosoma. So that was quite surprising. I thought, wow, that's interesting. I have no idea why that is the case. At first, it clearly falsifies this idea that it's just replicated often in textbooks about parasitoids, that is just said without any real hypothesis testing. And I started to think, well, why is that? And I started to think that maybe it's just a reflection of higher rates of overall diversification. Maybe coinobionts, because they're so much more specialized. Once a lineage becomes coinobiont, they speciate more and they generate more species. And just by virtue of generating more species, because even if each species is just a little bit different from the other, you have more morphological diversification. And so what John did was he used some of the current diversification and estimations of diversification methods like BAM and HISSE to estimate diversification in ichneumonids and see if that was linked to coinobionts and idiobionts. And that's what we saw is that, yes, many of the groups in which diversification is higher, here deep blue, the darker blue means slower rates of diversification, and lighter blue and red means higher rates of diversification. So most of these groups that have highest rates of diversification are indeed coinobionts. So it seems that, that this might actually make sense. I asked John then to make a linear regression, a correlation between rates of morphological diversification and rates of species diversification. And there is a statistically significant correlation, but as you can see, it's not something clear cut from the graph. There's a little line here, but there's plenty of noise in the data. So it seems that there's overall high support for this hypothesis that coinobionts change more, both in diversification rates and consequently in morphological diversification. But there's clearly a lot still to investigate here. So as we sequence more species and put more species in the tree and add more data, I think we'll be able to get a better grip on what's going on in this group. But I wanted to cite this just as an example of the kinds of things that we can start looking at with essentially the same kinds of data that we collect as systematists, in our everyday jobs. Another element of my work that I'll briefly mention, it's another expansion of my research program that I did as I was trying to make room for bigger evolutionary questions, interesting things, is studying genomics. So I was interested in sequencing whole genomes and using that to study biological adaptations and things about the ecology of each group. I will actually have a separate talk on this on Thursday. I think this will still be announced, or it was announced, I don't know. So I'm just going to talk a little bit here. It's just a trailer of what we're going to talk about on Thursday. But I just wanted to mention, just in case some of you don't make it to Thursday. So essentially, I'm using genomes to study the evolution of viral symbionts in ichneumonids. So as I was discussing, many ichneumonids are coinobiont parasitoids. So the host continues to feed and develop. So you see here the size of this wasp. You see how it's ovipositing in a tiny caterpillar. And this tiny caterpillar will be all of the foods that the wasp larva is going to eat all of their lives. So the reason why this wasp can oviposit in a tiny caterpillar and still be enough food is because the caterpillar also keeps eating, right? So the wasp larva is going to develop in the caterpillar while it's still active and eating. But of course, insects don't like to be slowly eaten alive. So they also have developed, mechanisms of defense against parasitoids. So they have immune systems that will fight the parasitoid. One of the things they have is defense cells, similar to our white blood cells that attack pathogens. And what they can do is that they can encapsulate the egg, circle it, and kill it, essentially destroy, suffocate the egg of the wasp. And so in turn, the wasp also needs to develop mechanisms to counter the immune system of the host, this kind of arms race between parasite and host. And one of the things that some groups of wasps do is they develop this, what we call nowadays, domesticated viral elements or endogenous viral elements, DVE for short, which are genes of viral origin that are integrated into the wasp DNA. So these are viruses that became associated so closely with the wasp that the virus genome was incorporated into the wasp genome. And so the virus is produced inside, all these viral particles are produced inside of the body of the wasp. And when it lays the egg in the host, it also injects these viruses, and these viruses will, in the host, they will express genes that will help to destroy the host immune system or modify its development to benefit the wasp. So again, I'm just going to mention this briefly today, but what's interesting is that, we know that this exists in some groups of parasitoid wasps, but we don't know exactly in which ones, because, finding these things, of course, as you might imagine, is complicated. One way of checking for the presence of domesticated viral elements is to dissect the wasp, take the wasp alive, dissect it, do electronic microscopy and see if you see viral particles there in the ovaries and so on. Obviously, that is very tricky to do with live wasps and identify them in the field, etc. Another thing you can do is you can try to isolate the virus with some techniques with proteomics, transcriptomics, preparation of cDNA libraries from the ovary of the wasp to see if you have viral genes being expressed in the ovaries and so on. So all of that is complicated. It requires being very good with experimental techniques and etc. But nowadays, the technological advances allow us also to use a brute force approach. Instead of all these elegant lab experiments, just sequence the whole genome of the wasp and see if you find the viral genes there. And that's what we've been doing at this point. Before, this would be very expensive and unfeasible, but now with genomics costs decreasing so much, we can start to look at these things in this kind of more straightforward way. So this is a project that I've been doing that is combining phylogenies and whole genome sequencing to try to understand the evolution of these viral associations in ichneumonidae. That is, in which lineages of ichneumonidae we see these viruses, how many times we think that these evolved, which groups of viruses were recruited, which original groups of viruses were originally incorporated into the wasp genome, and so on. This started some time ago when we sequenced the genome of 2 specific wasps. I work in collaboration with some French colleagues, and Nathalie Volkov, Fabrice Léger, and Stéphanie Robin. And we published a paper in which we sequenced 2 species of 2 more or less closely related species, and kind of mapped in their genome the location of the viral insertions in the genome and tried to understand the architecture of these of the structure of the viral genome inside of the wasp genome. But now what I'm doing is looking at the more phylogenetic approach. I'm looking at different groups of wasps and trying to see on which ones we can screen and find the genome. So that has been involving a lot of genome sequencing. So far, we sequenced 114 species with short read technology with Illumina at 20 to 60x coverage. And for 8 species, I also did Oxford Nanopore, which gives you really long reads and the possibility to assemble more complete genomes. And so, I'm going to talk about it more closely in more detail on Thursday. But what we're finding is that, we're being able to map. These are 2 different groups of viruses that have been integrated into the wasp genome. And we've been able to map those across the tree of ichneumonids. These are 2 specific subfamilies that were known to have viruses. And it was just generally assumed that, because we find them in these subfamilies, we're going to find it in all species of the subfamily. And what we're finding is that it's not exactly like that. So this subfamily here, Bankini, we found the viruses in most of the species, but not in all of them. So, like the Bancus vitosus, which is sister to all other species that we investigated, doesn't have the virus. So, this, as we add more and more species to the tree, is going to enable us to pinpoint the exact moment where this viral association occurred in the phylogeny. And in Campoplegine, which is another subfamily that also had the viruses, these purple circles here represent a viral association that was well known, and it was thought to be in all of the subfamily. But as we see, there's 2 major clades in the group, and only one of them has these viruses. So all of these other ones here don't have them. Instead, they have a second kind of virus that was known previously only from 1 species of Campoplegine, and it was thought this was kind of like a one-off event. But we find that it's actually present in several different genera. So, we're talking about 2 big evolutionary events of domestication of viruses in parasitic wasps. And again, what's interesting here as it relates to my work as a systematist, as a taxonomist, is that it's not so easy to identify all these species and have a phylogeny for them, right? You can, these people with whom I work, the French people who work on the genomics aspects of the viruses, they're very good with genomics, they're very good with bioinformatics, they're very good with viruses. But this is the kind of project that you can only do if you have a taxonomist on board, because you need someone to be able to collect these things, identify them, know what they are, select the proper species in the phylogenetic distribution, so that you can actually do comparative analysis across a broad lineage. If you don't have someone who understands the taxonomy of the group, then you just can't do this kind of thing. So this is a way of kind of combining these two very different expertises, people who are very good with the lab, very good with bioinformatics, and us who are systematists or taxonomists, but are interested in looking at interesting phenomena in a comparative light. Oh, okay, never mind, I explained this, but yeah, these are just some notes. What's also interesting in this my adventures in genomics is realizing that again, this can have a feedback on our systematics work, because now we're getting to the point where genome sequencing is becoming cheap enough that for many groups, especially if their genomes are small, we can start to do phylogenomics with directly sequencing whole genomes instead of doing UCEs, which you have to enrich your libraries, enrich your preparations to extract specific loci, or doing PCR where we go for specific genes. We can actually do shallow, low coverage genomic sequencing and then extract UCEs or whatever loci, whatever markers you want to use for phylogenomics bioinformatically, And with that, of course, we save a lot of time, we save a lot of steps, and at the same time, we generate data that can be used for other things as well. So you combine the sort of study of genomic architecture or gene evolution, whatever you want to do, with the possibility of doing phylogenomics for these things. In prices, they're still not so outrageous compared to the other techniques, the other genomic techniques. So again, this is another example of how these two worlds can communicate. Yeah, so this in short was kind of like an overview of how starting as a systematist and working on things that you normally work and generating data that you normally generate, we can start to look at different things, at projects and questions that definitely make it easier for us to talk to a broader audience, to publish papers in journals that are broader interest, and of course for those of us who are looking for jobs and that kind of thing, to get jobs and to get grants and so on. But at the same time, it's important, I think, to not lose sight of, if we're interested in systematics because we like it and we don't want to just abandon systematics. So my sort of like hopeful and optimistic view of this is that we can actually, do both. We're interested in now I'm starting this new job in Berlin in a department that's called the Center for Integrative Biodiversity Discovery, in which we're interested in how can we make taxonomy big science, the kind of thing that will attract the funding that is now available for phylogenomics and other big projects. How can we bring that to taxonomy? That is, the way I look at it, it's that we need to change the mentality of just taxonomy as a tool for providing names for species to a system for organizing all information about biodiversity. So bringing this omics mentality into taxonomy, making it taxonomics, something like that. Because, there's some things I realized in this work as I was, coming from a taxonomic background and working on other evolutionary questions, is that taxonomists are often the people with the first-hand information on things, right? The first ever things that we learn about a species are in a species description by definition, right? So those are the very first information that are available for a species. And for our insects and other neglected groups, they're often the only information that we have. All that we have for 90% of the species on Earth is a little list of morphological characters. So sometimes we are the people who have the only data available. We often have data that no one else has. So we often have a record, access to host records, to information on distribution, on phenology, temporal distribution, all these things that are also only available to us because they're still in a drawer in a collection somewhere and nobody has even published them. And all of this is especially true, again, for our insects and other poorly known organisms, which people are starting to realize now make up most of the biodiversity, right? So here is an estimation of the amount of biomass represented by different groups of organisms on Earth, right? We have like arthropods here, we have fish, annelids, mollusks, and this. And then here, we have wild mammals and wild birds, right? But I think all of us know that these groups receive, we spend as a scientific community, we spend a lot of time studying this and not enough time studying this, right? So if we want to answer big questions, if we want to understand big things about ecology and evolutionary biology, we need to broaden the scope of the things that we look at, just from this to all of this. And that requires going to the taxonomists and going to the people who have the only data that often we can look at. So, you know, and again, what kind of data are these, and how these can integrate a workflow in which we can both answer interesting evolutionary questions and advance systematics. So I started to think about, for example, things that I do for my normal work, my workflow, right? Things that I do in my day-to-day work. So usually we have a specimen, right? If I want to look at it in different ways, I'll do a DNA extraction and also keep a phenotypic voucher. Right. So we have the specimen, we have the DNA extraction. From the phenotypic voucher, we will take images and phenotypic descriptors. There is some characters, right? Measurements, whatever it is. From the DNA extraction, we can do, for example, DNA barcoding. DNA barcoding will help us make clusters of putative molecular operation taxonomic units, right? What we call bins. And then we can use the bins and the phenotypic descriptors, the characters, to delimit species, to understand among the specimens that we're looking at, which ones comprise the species based on genetic data and morphological data. Then we can also, we have these bins, we have the putative species. For each of these, we can generate genomic data. We're not going to generate genomic data for every single specimen, but we can generate one per species or per cluster, per barcoding cluster. Right, and from the genomic data, we have phylogenetic trees that we're going to use for a bunch of different things. We can do genome characterization, like for example, the studies I was just mentioning. Right, when we combine phylogenetic trees with the phenotype, we can do form to functions data sets with geometric morphometrics and investigations of ecological traits, what I was just mentioning. Right, so we have all these different things that are part of, like, at least my normal research. But when we look at all of this, what do we have? We have specimens, images, the characters, phylogenetic information and genetic information. I mean, that's the species description, right? That is centralizing all of the information about a species that we have in a single place. And that's what's interesting. That's what I think people are starting to realize. It makes having taxonomy and having species names an important thing, which is species names are nodes of data aggregation and distribution. Right. So what we have in practice is that we have one binomen, right? One Latin name, one species name that will integrate all of the information that we have about this taxon in a single unity. And so once we start to develop, use technology and a different mentality to kind of integrate all these data together so that they are really all in a single place, we can start to play with these data and generate things that are useful for research and for planning, conservation planning, etc. Right here we have, for example, species names associated with all this information. You have distribution records, you have phylogenetic context, you have images, etc. So, for example, if we take only the distribution records, we can make a checklist, we can make distribution maps, assessments of diversity in different areas. If we get, for example, phenotypic descriptors, that is characters and images, we can make an identification key. Or if we get, for example, DNA barcodes, we can do DNA barcoding reference library if that is associated with the species name. So, we can start to build all these products that are of interest to the society. People, conservation planners and decision makers are dying to get real comprehensive identification tools, really comprehensive checklists and information about where species are. So what we need is to kind of develop this mentality of integrating all this information together. And nowadays we have the technology to greatly accelerate our work. So these are some things that are being developed at the Center for Integrative Biodiversity Discovery in Berlin. I'm not developing them. These are my colleagues. It's under the leadership of my boss, Rudolf Meyer, but I thought it'd be interesting to mention them here. For example, we have this robot here, the diversity scanner, which you can load in a sample of a Malaise trap or something like that, and it will automatically go to each specimen and photograph it, and then use a pipette tip to pick it up and put it in a plate for DNA barcoding. That way you have the photos already associated with the specific location where it is in the plate. Then we can have the photo associated with the barcode sequence directly. So for example, the taxonomist can then receive in your office, before you even see any specimens, you receive a neighbor joining tree in which you can see the clusters of species of specimens and a photo for each one. So you can say, okay, I actually want to mount only these specimens. I don't need to mount 100,000 ichneumonids. I need to mount 2 or 3 per cluster. You have, for example, AI and machine learning techniques being developed that help you to automatically identify specimens. So again, at the center in Berlin, the diversity scanner can already identify many families using just the photos that it makes by AI with accuracy of about 91%, 100% for some groups. And so we can actually, in this process here, instead of having, for example, a single barcoding plate here, we could have in the future several. And then for each family, we load them in, the machine loads the specimens in one plate so that we can, only work directly with the groups of interest. The machine can also measure the specimens so that we can have direct information, approximate information, of course, on, how much of our sample is composed of small specimens, of big specimens, things that nowadays are super difficult to quantify, super difficult to assess, but it can be incorporated automatically and you don't even need to, think about it. You just get that information from your sample processing. And also with next generation sequencing, there's a lot of options for barcoding, but at really low costs. Now we're at the point where we can, using nanopore, for example, technology, you can generate barcodes for about 10 cents of a dollar per specimen. And that really gives us the possibility of actually barcoding every single specimen that we collect. Imagine that the barcode is the price of an entomological pin, right? If we can pin every specimen, we can also barcode every specimen. And then with that, again, we get to the point where not necessarily we're going to use the barcode as the sole tool for describing species, but we can use them as a pre-screening tool for that. If you have 300 specimens that have exactly the same sequence, you don't need to mount, pin, and label each one of them and then look at them at the microscope, because you already know they're genetically identical, at least for that gene. So I think that with all these things incorporated, we can look at a brighter future where instead of, resorting to the philosophy where the only thing to do is minimalistic taxonomic revisions where one barcode equals one species, we can think about maximalist taxonomic revisions, maximalist biodiversity research, where we really incorporate all of the information available into our workflow pipelines. And in the process, still get jobs and grants, hopefully. So yeah, muchas gracias. And I'll take any questions if I still have time.
¿Preguntas? ¿Alguien tiene alguna pregunta?
I have 2 questions. First, when you generate the morphospace, that's generated from the principal components analysis.
Yes.
From your map of the. So presumably, then the little map of points from the image, you're generating multiple characters from that image. Is that right?
The way that GM works is not really, it doesn't think of characters. It's just like the ensemble, the total configuration of landmarks is kind of like a single character.
Okay.
It's just one thing. The whole configuration is one thing. So if you would, for example, make several photos, you could say like the head is one character and the mesosoma is one character. There is actually something that has complicated, for example, efforts for inferring phylogenies directly from shape. There are people who are developing techniques to, for example, do phylogenetic reconstruction from shapes, but it's not super straightforward because each configuration is a single character. So you need to kind of like develop ways of modeling or reconstructing the evolution of ancestral shapes and so on.
Yeah, I guess I'm sort of confused about how you get from that sort of map of points on the specimen to the principal components analysis, because that requires the multiple characters to sort of build this.
Yeah, essentially what you will have is after you align your points, you will have that you can transform them into a 2-dimensional array in which essentially each specimen is a line, and because each landmark is going to be X and Y coordinates, right? As if it was like a map. So the X coordinate is a column, the Y coordinate is another for landmark 1. And, so you're going to have a number of columns or dimensions that will be equal to your number of landmarks times 2.
I understand.
So that's how it becomes like multivariate
Yeah.
data.
And my second question is when you saw that the rate of evolution for within the morphous space, that the rate of morphological evolution wasn't very high, but the rate of sequence evolution was high. Is that right?
Or in what part?
In the same part where you're using this morphospaces
Yeah.
to categorize morphological variation within the whole phylogeny, and you have the rate of morphological evolution
Yes.
was quite small, but the amount of
No, it was not it was not the amount of molecular evolution. It was the you had low diversity in shapes. That is, like, they occupied only a small part of the morphospace. But when you look at the phylomorphospace, the branch lengths are not molecular. They're morphological branch lengths. So these are like how different, like, for example, if you have two sister species, but they're here in different parts of the morphospace, you're going to have like branch lengths that connect them.
okay.
Right, that represent the degree of the difference between them. So what I was looking at was this, like, they occupied a smaller area of the morphospace, but when you look at the morphometric branch lengths, that is the amount of difference that you saw between each species, it was not inferred because of the relationships. It was not smaller than in the others. So the conclusion was that they were changing a lot, but only within a certain area of the morphospace. They could only change into certain morphologies, right? So you could change from one thing to here, but not to here. So you could like change from here to here, then back to here, but not to this other part of the morphospace.
¿Alguien más tiene preguntas aquí? Si no, damos paso a las preguntas en YouTube.
When using morphometrics, do you have any expectations about how they should look like in the morphospace before using them as characters for phylogenetic inference?
Can you repeat? Sorry.
Okay. When using morphometrics, do you have any expectations about how they should look like in the morphospace before using them as characters for phylogenetic inference?
Yeah. So I don't usually use morphometrics as characters for phylogenetic inference. So the phylogeny will come from molecular data. So the phylogenies will be derived from molecular data, and then the morphometric data will be used to plot that into the morphospace and see the differences between different species. Thinking about how they're going to look at the morphospace, I'm not sure I understand what that means, but I think one of the things you want to make sure of is that, you're choosing character systems and landmarks that have enough diversity. If you, for example, there are some things in the bodies of ichneumonids that are biologically interesting, but they're quite difficult to capture. For example, the abdomen. It's composed of several segments that are kind of interconnected and they slide onto each other. So you cannot really with one photo quantify them. So, for example, you could get one segment of the abdomen, but then it's just kind of like a trap, like kind of like a trapezoidal shape, and it doesn't have that much information. You'll be able to put maybe 4 landmarks there. So you have to juggle these things, like what areas are biologically interesting and at the same time are suitable for getting interesting landmarks. This is a limitation of doing work with photos. One of the things, of course, that lots of people are doing nowadays, it's using CT scanning, micro CT scanning to generate these 3D models, and then you can do much more sophisticated analysis, because then even like a leg, which is a relatively simple, structure, you can place landmarks across the whole leg in the 3D dimension, in the 3 dimensions, and then you can have a much better assessment of the shape of the leg.
Why would you expect branch lengths informs you about what is happening in other parts of the genome?
Well, again, in this case, the branch lengths I was talking about, they were morphometric branch lengths, not molecular. But I'm also interested in molecular branch lengths. For example, one of the things we did in that study was to correlate morphological branch lengths with molecular branch lengths to see if we were the groups that were changing the most in morphology were also changing the most molecularly, and that was the case. I think that sometimes branch lengths can be limited indicators of what's going on in the rest of the genome, for sure, because sometimes you're only dealing with like 4 or 5 genes, right? And so the branch lengths are not necessarily indicative of all of the change that's happening in the whole genome. But this is getting better, right? As we do things like UCEs, where you're sampling 2,000 different areas of the genome, then you're getting a much better estimation, I think, of how much change you have in between, across the phylogeny.
Last one. Is there any manual visual human checkpoint to verify all classification on the automated pipeline?
Well, you could insert them at any point you want. It's always a matter of how much time you have, how much manpower you have to look at these things. It depends on the pipeline that you're building, I guess. You could, you don't want to verify every single specimen, right? So, but you can definitely insert points where a human person checks, for example, a sample of the classifications, and then sees what is the level of success. And also, because everything is being barcoded anyways, you can always at a later time, if something was classified as, say, an ichneummonid, but is actually an ant, eventually you're going to get that barcoding sequence that's going to tell you that that's an ant. So I think there's ways of correcting the mistakes downstream the pipeline. Obviously, if the rate of success is so slow that if the rate of success of the algorithm becomes too low, then you spend more time correcting the mistakes than doing the work. But if you have a high enough success rate, then you start just correcting the mistakes start being not that much of a problem compared to dealing with the whole assembly of specimens. Yes.
Yeah. Thinking about this integrative taxonomy, have you or has anyone thought about including development data, either from genomics or transcriptomics, and correlating it with these morphometrics studies that you are developing? Or
Developmental data?
Yeah.
Huh. No, I'm not aware of anyone who has been doing that, but it would definitely be interesting. With these holometabolous insects, we often don't think enough about the, for example, larval stages, which are often the most important parts of the lives of these insects, and it'll definitely be good to be able to integrate them more seamlessly. I think when we get to the point where you're barcoding all specimens, you can also start to more easily integrating, kind of associating, for example, caterpillars with butterflies, because then you have DNA data for both, and then you can start to the more painstaking work. But I think it's also very interesting is to, once you associate larva with the adult, you can start to look what kinds of changes, if you have consistent changes between larva and adult, that can be used to then identify the larvae and so on. I have a friend who just published a work in which he reared mantid species, several of them from the egg until the adult, and then for each stage, for each instar, that he would fix and preserve one specimen, and then look at the whole transformation series across the development in several different species. And he was able to find several points in common that happened in the development of mantids. They are conserved in several species, so that you can establish, start to understand what's going on in their development, and because for so many of those groups, you cannot identify the nymphs or the larvae and so on. So once you start to find these consistent changes, then you can start to actually develop taxonomic identification tools for the other stages as well.
Cool, thank you.
¿Qué más tiene preguntas? Sí.
I was just wondering, for whole genome sequences, what methods are you using for phylogenetic reconstruction? Just concatenation or?
Yes, yes. In this case, yes, I was just doing concatenations. Sometimes, it's useful to do also coalescent species tree methods. But one thing I've found with UCEs is that often, because each UCE is pretty short, it's a pretty short sequence. Often, the gene trees that you get are not very good. You don't get really good gene tree estimations from each individual locus because each locus is often too short. So sometimes species tree methods are not perhaps so indicated for this kind of data, but other people have used them as well. It depends, I think, on the nature of your alignment.
And what about like whole genome sequences?
Yeah, with whole genome sequences, then you definitely could do it more easily. So with whole genome sequences, for example, you could extract buscos, which are single copy orthologs that are present, for example, in all insects or in all Hymenoptera and so on. And then you can get bigger sequences, and in that case, it should be easy should be more appropriate to do coalescent methods.
Thank you.
¿Tenemos más preguntas? Si no tenemos, bueno, sí hay una más en YouTube.
Have you seen or worked with geometric morphometrics data applied in different groups of animals or even plants? To answer these questions with the same approach, form to function.
Yes, there are plenty of other studies. In fact, my study with the phylomorph space was heavily inspired by a study by Brian Sidlauskas, who did a study on fishes, where he was investigating similar questions, also with the phylomorph. I think he was the one who developed first the phylomorphospace approach, but it was also looking at rates of evolution in two different groups of fishes. And yeah, there's plenty of other studies. Maybe not as many as it could. Maybe more people could get on board with that. But yeah, there's studies with other groups as well, linking phenotype with diversification or with ecological traits and so on.
Bueno, te damos las gracias otra vez, Bernardo. Muchas gracias por la plática.
Sí.
Y les comunicamos que tenemos café aquí en la parte exterior
Gracias.
para quien guste.
Bien, certificado. Gracias.